Bacterial genetic recombination. Bacterial genome, the genetic material of bacteria is DNA. In most cases, the entire genome of a bacterium consists of a single circular DNA duplex, often called the bacterial chromosome. It is located in ribosome-free cytoplasmic region called nucleoid or nuclear region. The genetic material of bacteria is DNA. We just said that. In most cases, the entire genome of a bacterium consists of a single circular DNA duplex known as bacterial chromosome. It is located in a ribosome-free cytoplasmic region called nucleoid or nuclear region. That region corresponds to the nucleus of eukaryotes. Still, it is not delimited by nuclear envelope and does not contain a nucleolus. Since the bacterial cell has only an unpaired chromosome, as in gametes, it is often regarded as a haploid cell. Its genes are neither dominant nor recessive, since all of them will be phenotypically expressed so long as there is no interaction between them. Plasmids and episomes. In addition to the main chromosome, many bacteria contain accessory extra chromosomal double helical DNA circles or mini chromosomes. They include plasmid and episomes. They contain extra chromosomal genes which provide some additional information that may be essential in certain environments. Plasmid are the free DNA circles which remain distinctly separate from the main chromosome. They replicate independently within the bacterium and get transmitted or inherited in a regular manner. They may get transferred from one bacterium to another during conjugation. Episomes are in fact integrated plasmids which remain incorporated with the bacterial chromosomes. So they replicate and get transmitted as a part of the main chromosome. Just like phages, plasmids also depend heavily on the metabolic machinery of the host cell for their replication, although each of them has its own genes and regulatory sites. Normally, they make use of the replication machinery of the host cell. However, each type of plasmid has its own genes for regulating the time of its multiplication and the number of its copies per cell. The number of copies per cell is usually specific for each kind of plasmid. Based on this number, plasmids are classified into low-key number plasmids or stringent plasmids with less than 10 copies and high copy number or relaxed plasmids, multiple copy plasmids with 10 to 100 copies. The specificity in the copy number of plasmids is maintained by the careful regulation of their segregation and distribution during cell division. This in turn is believed to be due to plasmid chromosome association, the details of which are yet to be understood. Then plasmid genes. Plasmids carry several sets of genes. The important ones among them are the following, like replication genes, which govern the replication of plasmids, incompatibility genes, which specify plasmid incompatibility. Plasmid incompatibility is the phenomenon in which plasmids carrying similar or identical genes cannot coexist in the same cell. Transfer genes, TRA genes, which govern conjugation and the transfer of genetic material from one bacterium to another. Resistance genes, which confer on the bacterium resistance against drugs, antibiotics, and antibacterial toxins. The genes which direct the synthesis of enzymes of specific metabolic function, nitrogen-fixing genes. For example, plasmids, episomes, and bacterial chromosomes contain transposons and insertion sequences or IS, which can move about both within and between chromosomes and plasmids and mediate genetic recombination between non-homologous sequences. So insertion sequences and transposons control the transposition of resistance genes. This in turn may generate plasmids which carry resistance genes. What is the significance of plasmids? Though plasmids are heritable self-replicating units, most of them are not indispensable for normal metabolism. Still, they are significant in the following respects. Plasmids carry the genes for developing resistance against antibiotics, drugs, toxins, and UV radiations, for controlling metabolism, and also for the production of bacterial toxins. Plasmids play an active role in genetic recombinations and also in the transmission of genes from one bacterium to another. Plasmids provide a very rapid method of gene transfer to meet the demands of the rapidly changing environment. Plasmids have very great practical significance to be used as cloning vehicles for the cloning or amplification of genes in recombinant DNA technology, genetic engineering. Apart from this, the conversion of milk to cheese and the production of some restriction and modification enzymes are governed by plasmid genes. Some plasmids can even cause disease in plants and animals. Then, kinds of plasmids. There are four major kinds of bacterial plasmids, namely F plasmids, F factors, fertility factors or sex factors, R plasmids, R factors or resistance factors, col plasmids, col factors or cholinogenic factors, and metabolic or degradative plasmids. Then F plasmids. F plasmids are the plasmids whose genes confer on the bacterium the ability for the transmission of genetic material from one strain of bacterium to another. They can have as plasmids as well as episomes, since they can exist and replicate both in the free and integrated states. The bacterial cells with the free F factors are called F positive cells, and those with integrated F factors are called high frequency recombinant HFR cells. F factors are actively involved in conjugation, so they are also called conjugative plasmids. F factors carry three sets of genes, namely replication genes, transfer genes, and incompatibility genes. The first set governs plasmid replication, the second set governs bacterial conjugation and genetic transmission, and the third set determines plasmid incompatibility. Of this, transfer genes are more numerous than the others. In bacterial cells possessing incompatible identical F plasmids will have similar or identical conjugation systems with little or no homology in their DNA sequences. Yet, all the cells having F plasmids, F positive cells, possess the following common features: the presence of sex spelling, the presence of transfer mechanism for the transmission of a copy of F plasmid to a recipient bacterium, the possession of surface exclusion property. Surface exclusion is the ability of an F positive bacterium to exclude or prevent the entry of an incompatible plasmid into it. This is accomplished by the activity of certain surface proteins called surface exclusion proteins. Then are plasmids. These are the plasmids whose genes confer on the bacterium resistance against the bactericidal or bacteriolytic effects of fungal antibiotics, drugs, etc. An R plasmid has two contiguous parts, namely resistance determinant factor or R determinant and resistance transfer factor RTF. The former contains the genes for resistance against antibiotics and drugs, R genes, and later carries the transfer gene which govern the transmission of resistance from one strain of bacterium to another, and also the genes which regulate DNA replication and the copy number of plasmids. Resistance transfer is mainly through the activity of transposons. Small drug-resistant plasmids without transfer genes are also common. R transfer differ from F transfer factors in that they cannot get integrated with bacterial chromosome to serve as episomes. A significant feature of R plasmids is their high transferability. They can be readily transferred to any other bacterium, irrespective of the strain or species of the recipient. High transferability enables R plasmids to donate resistance to all bacterial cells in the neighborhood. In other words, R factors enable bacteria to acquire resistance against drugs and toxins and thereby to attain high ecological advantage and survival value. This in turn is a serious threat to public health. Bacterial cells containing R plasmids are generally immune to superinfection. Then col plasmids. These are the plasmids whose genes are responsible for the production of certain bacterial toxins known as bacteriocins. Bacteriocins produced by one strain of bacterium can bind with the cell wall of others and get into them. They can inhibit gene replication, transcription, translation, energy metabolism, and growth of the recipient, or even kill them. Colsins produced by E. coli and some other enteric bacteria. Vibriosins produced by Vibrio cholerae, the cholerae bacterium, etc. are bacteriocins. A bacterium is resistant immune to its own toxins, though it is susceptible to a different variety. This protection is afforded by a protein toxin complexing which inactivates the toxins. When this complex happens to attach to the surface receptors of a susceptible strain of bacterium, the immunity protein dissociates from the complex and the toxin penetrates into the recipient cell. Just as F factors, cold factors also can serve as episomes. At the same time, they resemble bacteriophages in that they can bring about the death of the recipient bacteria. Then metabolic plasmids. Metabolic or degradative plasmids are the plasmids whose genes govern the production of metabolic enzymes. In the soil bacterium Pseudomonas, the production of the enzymes for the breakdown of phenol, hexane, xylene, camphor, etc. is governed by plasmid genes. Metabolic plasmids can be transmitted through conjugation together with F factor. Then embryonic plasmids. Plasmids have been detected less commonly in eukaryotes also. The most common and important one among them is the yeast plasmid killer particle. Not that much common, but it's very important. It is a double-stranded RNA molecule and the only known plasmid which does not contain DNA. It contains 10 genes for replication and many others to govern the synthesis of a bacteriocin-like killer substance. As in bacteria, most eukaryotic plasmids are circular, but a few isolated from maize, sorghum, and many fungi are linear DNA molecules. Then plasmid replication. Bacterial plasmids can replicate only within host cells. At the same time, there is a wide variation with regard to the enzymes and molecular mechanics of plasmid replication. Plasmids depend heavily on the replication machinery of the host cell for their replication. Some of them even make use of entire genome products of the host, whereas the others require some plasmid gene products. All known plasmids replicate semi-conservatively and maintain their circularity throughout the replication cycle. Still, the pattern of replication is different in different kinds of plasmids with regard to the directionality, termination site, etc. Replication is unidirectional in some plasmids, bidirectional in some others, and both these types are present in still others. In unidirectional plasmid replication, termination of replication occurs at the site of origin after each cycle, but in bidirectional replication, termination occurs after either at a fixed point away from the origin or at the meeting point of the two replication forks. In any case, no termination signal is known to occur. In most plasmids, replication occurs in the butterfly model. In this case, the replicated portion is untwisted and the non-replicating or resting portion is positively supercoiled. On the completion of the replication cycle, one of the DNA circle is cleaved, probably by gyrase activity. This results in the formation of a nicked molecule and supercoiled molecule. The nicked molecule soon gets sealed and later on becomes supercoiled. Then gene transfer and genetic recombination in bacteria. Next major topic. Bacteria reproduce only by amitotic fission. So they do not have any regular program for introducing genetic variability through sexual reproduction, meiosis, and crossing over. Yet they bring about gene shuffling in a rather unplanned way. They can produce new genotypes and phenotypes through mutation. Also, they can acquire the effects of sexuality, recombination, and genetic variability by importing genes from outside. The wild types and capable of undergoing mutation are called prototrophs, and the altered mutant forms are called auxotrophs. It is believed that bacterial DNA contains some mutant genes which can induce mutations in other genes. Bacteria accomplish gene transfer and recombination by four principal mechanisms, namely conjugation, transformation, transduction, and transposition. In any of these mechanisms, gene transfer is never mutual or reciprocal, but is always one-sided or unilateral. So it involves a one-way transfer of DNA from a donor cell to a recipient cell. Of the four mechanisms, the first three are parasexual processes because the donor bacterium and the recipient behave as male and female respectively. Moreover, the donor DNA and the recipient DNA will have homologous sequences. On the other hand, transposition does not require such a sequence homology and it involves the integration of transposable genetic elements. Then bacterial conjugation. Bacterial conjugation is the unidirectional transfer of genetic material from male-like donor bacterium to a female-like recipient bacterium of the same species through direct cell-to-cell contact. it is always the F factor that is transferred from the donor to the recipient. So conjugation always occurs between a F negative factor possessing or F positive donor and an F factor lacking or F negative recipient, and never between two F positive strains nor between two F negative strains. By receiving the F factor from the donor, an F negative recipient becomes an F plus donor. This mixing a population of F plus bacteria with F minus forms makes all forms F positive. Conjugation was first reported in E. coli by Lederberg and Tatum in 1946, the co-recipients of 1956 Nobel Prize. There are two types of bacterial conjugation, one between F positive donor and F negative recipient, and other between HFR donor and F negative recipient. Conjugation between F positive donor and F negative recipient. This is the commonest method of bacterial conjugation. It involves following four major events: effective contact and parasexual pairing, cell-to-cell attachment, and the formation of specific pairs between donors and recipients. Mobilization or preparation for the transfer of the donor DNA, transfer of donor DNA to the F negative recipient, then replication, formation of a functional replicative plasmid in the recipient by the synthesis of a complementary companion strand and the circularization of the DNA molecule. Many plasmids fail to carry out all the above processes. Accordingly, they are classified under three categories, namely conjugative plasmids, mobilizable plasmids, and self-transmissible plasmids. Conjugative plasmids carry the genes which govern the effective contact between the conjugates. Mobilizable plasmids carry the genes which govern the preparation for DNA transfer. Self-transmissible plasmids are both conjugative and mobilizable F plasmids. The process in which a non-conjugative plasmid is transferred through effective contact provided by the conjugative plasmid is called donation. A conjugative plasmid can also help the transfer of a non-mobilizable plasmid. In this case, there occurs a genetic recombination between them, forming a single transferable plasmid. This kind of genetic transmission through the recombination of plasmids is called conduction. It is relatively less frequent. Then effective contact and the formation of donor-recipient pairs. The first step in bacterial conjugation is the formation of a mating pair between an F positive donor and F negative recipient through cell-to-cell attachment. During this, hair-like appendages develop on the surface of donor cell. They are called sexpili. Their synthesis is governed by the F factor genes. Chemically, they are formed of proteins, carbohydrates, and phosphates. The protein part of pilus consists of a single peptide chain called pilin. Sexpili binds with receptors on the surface of F negative recipient cell. Soon they retract to the donor cell, bringing the mating cells into close pairing. Closely afterwards, one of the pili serves as a hollow intercellular bridge between the donor and the recipient for the transfer of the genetic material. It is called conjugation tube. Next process, mobilization. The direct cell-to-cell contact and the formation of the conjugation tube signals mobilization or preparation for DNA transport. During this, a single-strand nick is made by endonuclease in a specific base sequence in the F factor called transfer origin. This nick initiates a rolling circular replication of DNA. During this, the nicked strand separates from the circular companion strand and becomes linear. The 5' end soon gets complexed with the nicking protein or with other proteins. This complexing protects it from degradation by nucleases during the transfer. Simultaneously, the circular strand in the donor cell acts as a template and guides the synthesis of a complementary companion strand, forming a circular DNA duplex. This duplication of a factor strand in the donor cell is called donor conjugate DNA synthesis, DCDS in short. It occurs independent of the duplication of chromosomal DNA. So chromosomal DNA has no role in this process. It neither gets duplicated and integrated with a factor nor it gets transferred to the recipient cell. Transfer of the single-stranded donor DNA. The single-stranded and linear donor DNA soon moves to the recipient bacterium through the conjugation tube with its 5' end serving as a leading or advancing end. It is held that The entry of donor DNA into the recipient cell is facilitated by specific surface proteins of the recipient cell. The surface proteins are believed to interact with the pilot proteins at the advancing end of the donor DNA, with the result that the donor DNA is drawn into the recipient cell. Then replication. Replication is the conversion of linear single-stranded plasmid DNA transferred from the donor bacterium to double-stranded circular DNA in the recipient bacterium. So it involves two processes, namely replication and circularization of the single-stranded DNA. The replication of this linear DNA strand in the recipient cell is called recipient conjugative DNA synthesis, RCDS. During this, the single-stranded DNA serves as a template and guides the synthesis of a complementary companion strand. The linear DNA soon transforms to a circular closed circle. The cellular DNA thus formed makes the F negative recipient and F positive donor. Shortly afterwards, the two conjugates separate. The next topic is conjugation between HFR donor and F negative recipient, sexduction. There are instances of bacterial conjugation in which a part of chromosomal DNA also is transferred to the recipient cell. This is called sexduction or abduction. In this case, the F factor of a donor cell behaves as an episome. It gets integrated with the chromosomal DNA, forming a recombinant DNA called high-frequency recombinant DNA, HFR DNA. The frequency of this recombination and that of the subsequent conjugation is very high, so it is called high-frequency recombination. The bacterial strains possessing HFR DNA are generally called high-frequency recombination strains, HFR strains. They will not have free F plasmids. They were first isolated and named by Lederberg in 1952. HFR strains arise spontaneously with the F positive strains by the integration of F negative plasmids with chromosomal DNA. By a reverse process, F negative plasmids get dissociated and the HFR cell transforms to an F positive cell. In the integrated state, the autonomous replication machinery of the F negative plasmid ceases to function. At the same time, the plasmid DNA replicates passively as a part of the chromosomal DNA and gets transmitted to hundreds of progeny cells. Then, conjugation between HFR donor and F negative recipient is peculiar in that genetic transfer is governed by a transfer gene of the F negative factor. During this, cell-to-cell attachment and the formation of a conjugation tube occurs exactly as in the conjugation between F positive and F negative cells. Then the HFR DNA undergoes rolling circle replication. In this process, a single strand nick is made in a specific site within the integrated F negative plasmid by internuclease activity. This nicking site is called origin of transfer. The 5' end of nicked strand progressively separates from the circular companion strand. As a result, the nicked strand becomes linear. Since the nick is somewhere within the F factor, a portion of the F negative factor occupies the 5' end and the other portion occupies the 3' end of the linear strand. Now the 5' end behaves as a leading end and the 3' end as a lagging end. The leading end moves to the F negative recipient through the conjugation tube before the entire strand gets into the recipient cell. The conjugation tube and the DNA strand break off and the conjugates separate. As a result, a part of one strand of chromosomal DNA and a portion of the integrated F negative transfer factor gets transferred to the recipient cell. The single-stranded DNA transferred to the recipient now acts as a template and synthesizes on it a complementary companion strand. The double-stranded DNA thus formed soon gets integrated with the recipient chromosome. During this, homologous portion of the recipient chromosome endognode is sliced away by internuclease activity, and into that gap, the donor DNA exogenote is spliced with the help of DNA ligase. This results in the formation of a recombinant HFR chromosome in the recipient. Simultaneous with this, the lost part of the chromosome of the donor cell will be resynthesized. The site at which the F factor exogenote is incorporated with the bacterial chromosome exogenote is called insertion sequence. Conjugation between HFR donor and F negative recipient is significant in that, in addition to the plasmid genes, some chromosomal genes are also transferred to the recipient. This enhances very much genetic variation in the recipient system. In the conjugation between F positive and F negative cells, no chromosomal genes are transferred to the recipient. Then bacterial transformation. Transformation is the transfer of genetic material from one strain of bacterium to another by direct intake of DNA through a medium. It was discovered by Griffith in 1928. In transformation, DNA fragments are released to the medium from a dead and broken bacterial cell. They are called transforming molecules. They are soon taken in by another bacterium or active transport through certain specific points like receptor sites or entry sites. In the recipient cell, each fragment gets integrated with the chromosomal DNA by recombination and forms a recombinant chromosome. The uptake of transforming molecule is an active energy requiring enzymatic process, so it occurs only in those cells which possess the necessary enzymes. Steps in bacterial transformation: binding of the transforming molecule. In transformation, the double-stranded donor DNA fragments first bind with specific receptor sites, entry sites on the surface of recipient cell with the help of membrane-bound enzymes. Then next step, uptake. Each foreign DNA fragment gets cleaved into still smaller pieces by ribonuclease activity. These pieces are then taken into the recipient cell by active transport with the help of DNA translocase. Then degradation into single strand. Degradation of one strand of each transforming molecule by exonuclease activity takes place and only one intact strand gets into the cell. But in some cases, double-stranded fragments may be taken in by the recipient. One strand of each fragment soon gets digested by nuclease activity. In any case, the intact single strand remains in the recipient cell for some time, only for a few seconds. This period is called eclipse period. Then integration process. Then a part or whole of a single stranded donor DNA is inserted into a complementary or homologous part of recipient chromosome. During this, a homologous portion of one strand of recipient DNA is sliced away by endonuclease activity. And onto that gap, the donor strand is integrated with the help of DNA ligase. Since the donor strand and the recipient strand are not genetically similar, the double helical region formed by them is called heteroduplex. Then expression in the transformed recombinant cell, segregation and phenotypic expression of the integrated donor gene take place. Then in transformation, the double-stranded donor DNA fragment first bind with specific receptor sites on the surface of recipient cell with the help of membrane-bound enzymes. Soon, each fragment gets cleaved into still smaller pieces by endonuclease activity. These pieces are then taken into the recipient cell by active transport with the help of DNA translocase. The energy of this transport is derived by the simultaneous degradation of one strand of each transforming molecule by exonuclease activity. So only one intact strand gets into the cell, but in some cases, double strand fragments may be taken in by the recipient. One strand of each fragment soon gets digested by nuclease activity. In any case, the intact single strand maintains in the recipient cell for some time, only for a few seconds. This period is called eclipse period. Then it gets sliced or spliced to a complementary or homologous part of chromosomal DNA. During this, a homologous portion of one strand of recipient DNA is sliced away by endonuclease activity, and into that gap, the donor strand is integrated with the help of DNA ligases. Since the donor strand and the recipient strand are not genetically similar, the double helical region formed by them is called heteroduplex. Then expression, which we learned just now. Then competence. Not all bacterial cells can take in donor DNA and get transformed, but only some can. They are called competent cells. Their particular physiological fitness and ability to take in exogenous DNA and to undergo transformation is called competence. What makes a bacterial cell competent for transformation is not definitely known. Some environmental factors in the external medium known as competent factors are believed to be essential. They include some proteins and some thermostable molecules. They possibly make the cell surface ideal for the binding of the transforming molecule. In bacterial transformation, there is some sequence homology between the incoming exogenote and recipient endogenote. In the absence of such homology, transformation may not occur. This makes it clear that a bacterial cell can be transformed only by the strains or species of bacteria that are genetically related to it. It has been shown that the introduction of viral DNA into a bacterium results in the formation of an intact viral particle inside the bacterium. This is called transfection. In this case, the viral DNA does not actually get integrated with the bacterial DNA. When such a transfected bacterium infects an animal host, it releases the viral particle, and this causes viral infection of the animal host. Next point, bacterial transduction. This is the transfer of a part of genetic material from one strain of bacterium to another through a bacteriophage. In other words, it is the phage-mediated bacterial gene transfer. The carrier phage is called transducer or vector. Transduction was discovered by Joshua Lidenberg and Norton Center in 1952. It serves as a mechanism of bacterial recombination. It is now widely applied as a technique in recombinant DNA technology, genetic engineering. Transduction is of two kinds, namely generalized, common or random transduction, specialized or restricted transduction. In the first one, like generalized, any gene or any DNA segment can be transferred by the virus. But in the later, only certain specific genes or specific DNA segments are transferred. Generalized transduction is mediated by some lytic phages whose DNA does not get integrated with the bacterial chromosome. Examples are the P1, P22, PBS11 phages. On the other hand, specialized transduction is mediated by lysogenic phages whose DNA gets integrated with the bacterial chromosome. Then generalized transduction. In generalized transduction, a portion of the donor bacterial DNA gets enclosed by the capsid proteins of the phage, forming a transducing principle. This happens as a mistake in phage assembling at the end of the lytic cycle. In this case, the capsid protein gets organized not around the phage DNA but around the bacterial DNA fragment. This transducing particles are the viral particles with bacterial DNA. This transducing particle is released along with the viral particle by the lysis of the bacterium. It attaches to the surface of a new bacterial host and injects the DNA into it. Then reciprocal recombination occurs between the injected DNA segment and a homologous part of the bacterial chromosome, forming a recombinant DNA. Then specialized transduction. Specialized transduction is closely associated with the lysogenic cycle of temperate phages. In this case, the phage DNA gets integrated with the bacterial chromosome and forms a prophage. The prophage undergoes repeated replication as a part of bacterial chromosome. After that, it undergoes induction, during which the phage DNA gets extricated by cleavage exactly at its points of attachment with bacterial DNA. But sometimes this cleavage occurs erroneously a little away from the attachment site. As a result, a portion of the phage DNA is retained in bacterial DNA and a portion of bacterial DNA remains attached to the excised phage DNA. The excised phage DNA and a portion of the bacterial DNA attached to it forms a specialized transducing particle. This particle carries only those bacterial genes that are located close to the site of prophage insertion. Thus, it becomes clear that specialized transduction can transfer only the genes that are closely linked with the prophage. The specialized transducing principle is released by the lysis of the host bacterium. It infects a new bacterium and gets integrated with its chromosome through recombination. Thus, a specific chromosomal segment of the first bacterium gets integrated with the chromosome of the second bacterium. In other words, the second bacterium gets transduced. The transduced bacterium is often called transductant. Transductants having integrated transducible principle are generally called primary or unstable transductants. They are usually unstable because the transducing principle often gets extricated by introduction. The phage DNA of the excited particle is unable to replicate since it is incomplete. So it may get disintegrated through successive divisions of the host cell. On the other hand, the bacterial DNA of the particle often undergoes recombination with the host chromosome, transferring only some genes to the later. This produces a secondary or stable transductant. It is stable and very seldom it loses its exogenode through introduction. So in this session, we try to learn about genetics of bacteria and phages, conjugation in bacteria, transduction, generalized and specialized, sexduction, structure and life cycle of bacteriophage, which is explained with the help of a previous video, temperate and virulent phages, lysogen and lysis, that is also explained in another video. So thank you for listening.